All right, let's dive right in. Today, we're going to crack open the playbook that runs, well, pretty much every living thing, gene expression. You can think of it as the ultimate instruction manual for life.
So you've got this DNA, right? It's basically a massive, super detailed blueprint. I mean, it's got the plans for literally everything that makes you you. Your eyes, your hair, even the nitty-gritty of how your cells work. But a blueprint just sitting there doesn't build a house. It's useless if no one's reading it. So the real question is, how does our body take all that amazing information and actually do something with it?
Well, the secret is in these little things called genes. A gene is just a specific chunk of your DNA. A great way to think about it is like a single recipe in a giant, giant cookbook. And just like a recipe gives you the steps to make a specific dish, these genetic recipes are what build all our physical treats. And they do it through this really amazing process. And that process? That's what we call gene expression. It's the whole journey, really, from the raw information, the code that's locked away in a gene, all the way to a final, functional product. And usually that product is a protein, a little molecular machine that goes out and gets a specific job done in your body. Okay, so this whole thing basically breaks down into two main steps. First up, we have transcription. That's where we make a copy of the gene. And then there's translation, which is where that copy is used to actually build the protein. Let's stick with that factory analogy because it works really well here. You wouldn't take the master blueprint, the original copy, out onto the messy factory floor, right? No way. You'd make a photocopy first. And that's exactly what the cell does. All right, so let's look at step one, transcription. The cell calls in this special enzyme. It's like a molecular machine called RNA polymerase. Its whole job is to create a temporary copy of a gene. And this copy has a name, messenger RNA, or just mRNA for short. This is our photocopy. It's the working copy that can safely leave the head office, the cell's nucleus, and travel out to the workshop.
Okay, now for step two, translation. That mRNA copy, our photocopy, makes its way to the cell's factory floor. This is where the action happens. The machinery of the cell latches onto it, reads the instructions, and starts putting together the protein one little piece at a time exactly, and I mean exactly, as the original gene blueprint specified.
This can all feel a bit abstract, so let's make it real. Let's talk about keratin. You've probably heard of it. Keratin is the protein your body uses to build your hair, your fingernails, and the outer layer of your skin. It is the perfect, everyday example of gene expression in action. So there's the gene for keratin, and it holds the recipe. Simple enough. But what if the factory just went haywire? What if it just kept pumping out keratin nonstop, 24/7, with zero oversight? What happens if that instruction is just left on full blast all the time?
Well, let me tell you, the results would not be pretty. We're talking about things like way too much hair growth, skin that gets all dry and hard, and nails that are unusually thick and long. It's not a good look, and this just perfectly shows why having the instructions isn't the whole story. You absolutely have to have control.
And that control system has a name. It's called gene regulation. It's this whole set of complex mechanisms that basically acts like a factory manager. This manager is the one who decides which production lines get turned on, which ones get shut off, and, just as importantly, how much of each protein gets made. It is absolutely crucial for keeping the entire operation running smoothly and in balance. So this regulation is incredibly, incredibly precise. And to really get how it works, we got to zoom in and look at the language of the instructions themselves. How does the cell actually read this stuff? Well, it turns out that the language on that mRNA copy isn't just a jumble of letters. It's written in specific three-letter words, and these are called codons. And this genetic language, it's not just random. It has very strict rules. For starters, it's commaless. That means it's read continuously, back to back, with no spaces or punctuation. It's also non-overlapping, which means each genetic letter is only part of one three-letter word at a time. Super efficient. It also has polarity. It's always read in one direction, from start to finish, just like you'd read a sentence. And then there's this last one, which sounds kind of bad, but it's brilliant. It's degenerate. All that means is that several different three-letter words can actually code for the very same building block. This is a fantastic safety feature because it builds in some redundancy and helps protect against typos or errors. So here's a quick look at what that means in practice. The codon AUG, for example, is the universal signal that says start building here. Something like UCU tells the machinery to add a serine. And then you have codons like UAA, which are basically a big red stop sign. See? It's a really clear, precise language of instructions. Now, here's a couple more really important points. The code is non-ambiguous. That means one codon always stands for one specific amino acid. There's no guesswork. And here's the mind-blowing part. The code is pretty much universal. The same three-letter words mean the same thing in you, in a giant oak tree, in a little bacterium. This shared language is the whole reason something like genetic engineering is even possible. And on a deeper level, it's a huge, profound clue about the shared history of all life on this planet.
Okay, so the basic process is the same, but the factory layout can be a little different. In simpler cells, like bacteria, those are prokaryotes, everything's kind of mixed together. Transcription and translation happen at the same time, right there in the cytoplasm. But in more complex cells, like ours, we're eukaryotes, things are more organized. We have a separate head office, the nucleus. So transcription happens safely inside the nucleus, then the finished mRNA copy is shipped out to the cytoplasm for translation. It's a much more controlled, separated workflow.
So, when you boil this entire incredible process down, you get one fundamental rule. Information flows in one direction, from the genes to the proteins, from the DNA blueprint through that mRNA messenger to the final protein that actually does the work. This one-way flow of information is so important it's called the central dogma of molecular biology. It literally governs all of life as we know it.
And that brings me to a final thought I want to leave you with. Just stop and think about this for a second. The fact that you, a tiny bacterium, and a massive redwood tree are all reading from pretty much the exact same genetic playbook. I mean, that's just staggering, isn't it? So what does this shared ancient language really tell us about the deep, interconnected story of all life on Earth?